We're going to get started because I got a lot of material I got to cover. So today we're talking about epigenetics. That is the study of the ways in which changes such as like epigenetic traits can alter cell and tissue patterns of gene expression. So what do I mean by an epigenetic trait? Well, that's a stable, mitotically, and in some circumstances even meiotically heritable phenotype, especially as cells continue to divide, that results in changes in expression without alterations in the DNA sequence itself, right? DNA is made up of A, G, C's, and T's, but we can make chemical alterations to the DNA that doesn't change that, the A, G, C's, and T's, but does change whether things get expressed or not. And that's epigenetics. And this means that, like, okay, so we refer to things such as the epigenome, and that's the epigenetic state of a cell. And this means that over the course of a lifespan, one organism can have obviously just one genome. That doesn't change over the course of that organism's lifespan. But the genome can be modified in many diverse cell types at different times to produce many different epigenomes. And so the actual chemical structure will be different even if the A, G, C's, and T's is not changed. Gesundheit. Now, epigenetics has been implicated in a bunch of different things, and this is just a subset. So one of those things is the progressive restriction of gene expression during development. So early on in embryogenesis, when you have a single cell that gets produced, and then you've got the 2-cell stage, the 4-cell stage, there are many, many genes that are on during those phases. As cells become specialized and differentiate, we see a loss in the numbers of genes that are expressed. So we get a restriction of the kinds of genes that are expressed. And it is thought that epigenetics plays a major role in that restriction of the kinds of genes that get expressed as tissues differentiate and become specialized. It also plays a major role in allele-specific expression in gene imprinting, especially in those circumstances that violate what you might think would arise from typical Mendelian inheritance. And then there's also environmental genome interactions during prenatal development and postnatal development that can affect adult phenotypes. And I'm going to go through it thoroughly one very famous example of this. So when I talk about epigenetic alterations to the genome, I'm talking about 3 categories, okay? So methylation, which is something we've talked about, but we're going to spend a lot of time talking about today. These are changes that are reversible, and it includes the addition and removal of methyl groups to the genome. We'll be spending a lot of time talking about this. We've also talked about histone modification in the past and chromatin remodeling. This is going to affect the availability of genes for transcription. Again, something we've already discussed, so I won't spend a lot of time talking about this. But I am going to spend a fair amount of time talking about non-coding RNA. Okay, so we're going to dwell on that a little bit more. So DNA methylation. So I'm going to go into this in a little bit more detail. This is when you have enzymes known as DNA methyltransferases, or DNMTs. A methyltransferase will add a methyl group to cytosine, and this produces 5-methylcytosine. So cytosine is the C, right? It's the nitrogenous base that makes up the C's in their genome. And at the 5th carbon, there normally is a hydrogen attached. But if it's been methylated by a methyltransferase, well, then the hydrogen gets removed, and instead we have a methyl group that gets attached. This changes the basic sort of physical structure of this nitrogenous base, which means that this nucleotide has now changed just a little bit, but enough to have an effect. Okay, so it's when we talk about methylation, it is just happening on C's, but not just any kind of C. It has to happen on C's that are adjacent to G's. Okay, so we have to have a cytosine adjacent to a guanine, and this is referred to as a CpG dinucleotide. So it has to be C and then G in the genome, and that is referred to as the CpG dinucleotides. And this can be methylated. Now, what we find is that in promoter regions, those are the cis-acting elements adjacent to a gene that might be turned on, right? That is a promoter region, where you would have a transcription factor that would bind to it to allow for the gene to be transcribed that's downstream. Within those promoter regions, we often find what are referred to as CpG islands. So a CpG island is a portion within the promoter region where you have a lot of CG sites. And I took this from a figure from a paper where they have in bold all the CG sites that is just within one stretch of the whole CPG island, just to illustrate that you can have a bunch of these in place. And so here's a CG, here's a CG, and within this, relatively short sequence, we have 17 different CPG dinucleotides shown. So these are lots of places where we can have methylation occur. And yeah, we find this adjacent to all kinds of important genes. So the way your textbook illustrates this is that the CPG island will be in the promoter region, and they're showing it as like open circles means that it's unmethylated, and therefore the gene is turned on. But if a bunch of these CPGs in the CPG island are methylated, well, then the promoter can't really bind very well, and this will silence the downstream gene. So this is how you can get regulation of gene expression through chemical changes to the genome, but without actually changing the sequence, okay? And even though this is reversible, it's not easily reversible. This methylation can last days, weeks, months, even years. So a site can be methylated and it will last for a lifetime. It could, potentially. And you need to have demethylation occur. So there would have to be different enzymes to come in and remove those methyl groups. So these can be very long-lasting, largely permanent changes that then results in like massive consequences for that cell. So instead of regulating expression through, say, repressor proteins or promoters or transcription factors, right, that can turn it on and off, this is a way that's like, this is basically turned off more or less permanently unless we come in and we strip all these methyl groups off, okay? So it can have huge consequences. And then this is also from your textbook showing like where these CPG sites can look if we do this ball and stick type, the space filling model for DNA. This is what this is referred to, okay? So this is a DNA double helix, obviously. And these would be two CPG sites here. And as you can see, we've got two methyl groups that are here that they've illustrated in red. And with these added methyl groups, if it happens to be within the major groove of DNA, this is where you would typically then have the promoter site come in, and a transcription factor could bind in here. But if you had a bunch of these methyl groups that are sticking up like this, well, then that promoter won't bind very well to the DNA. So this is a way of adding in a bunch of junk that gets in the way, and that junk being methyl groups, so that it won't bind. And that's kind of how that works. Does that make sense? So you're physically altering the DNA so that it now can't bind. Okay. So it turns out that like methylated CPG dinucleotides are found in repetitive sequences, like especially within heterochromic regions. So the one reason why we refer to heterochromatin as not having much transcription that occurs is because it is highly methylated. That's one thing that makes heterochromatin its overall unique structure. And that heterochromic methylation actually offers some stability for those regions as well. So you're less likely to have translocations and other chromosome abnormalities if a region is highly methylated. So it does provide some additional stability. So not only do you get that stability, but it's also then going to be a portion of the chromosomes where you don't get a lot of expression because they're highly methylated, okay? Now, methylation also plays a role in X chromosomes, in activation, which we will talk about in a moment. And then it also plays a major role in imprinting as well, which we will also talk about in a moment. Okay, questions on methylation before I go on? All right. We'll be talking a lot more about methylation. We've also already talked about histone modification. So I've already done this before, but I'm going to go over it sort of briefly. That we know that we have condensed chromatin, and that chromatin can also be relaxed. And modifications to the histones is one way that we get that that either opening or closing of the chromatin to make the gene sites either available or unavailable. And this can happen through acetylation, methylation, or phosphorylation. So if we zoom in on this, we can see that we have acetylation sites, we've got methyl sites, we have got phosphate sites on the N-terminal ends of these different histones, right? So these N-terminal ends are sort of sticking out like this. And So you might add an acetyl group or a methyl group or a phosphate, and this will then change the conformation of the histone, which will either condense or relax the chromatin. Okay. Now, acetylation is done by histone acetyltransferase. If you have that enzyme active, it tends to acetylate the histone end tails. This will open up the chromosomes typically. And so therefore, the chromosomes are open in a relaxed state, making genes available for transcription and then translation. You can remove those acetyl groups from those histones by histone deacetylase, HDAC. And therefore, then it is found in a closed state. And a lot of times there's also sites along either DNA or potentially even the N-terminal ends that can be methylated in various ways. And methylation tends to make genes silenced to some degree. So these little black dots here, they're showing as sites of methylation. So this then leads to us to seeing that there's some kind of histone code, okay? So within the amino acid structure of the N-terminal end of the histone, so what we're illustrating here is just histone 3 and then histone 4. We can do the same thing for histones 2A and 2B if we wanted. But this is 3 and 4, that these are all known sites for modification that can occur. Some of it's methylation, some of it's acetylation, some of it is something called ubiquitination, so you can add a ubiquitin tag, which is another form of modification to the histones. And then this will then change those histones, the conformation of those histones, so that it'll either open or close the DNA, making it either more accessible or less accessible. And so our understanding of this code is that we can look for these sites and make determinations about whether genes are going to be more likely to be turned on or off, depending upon if they've been methylated or acetylated or ubiquitinated at particular places. Does that make sense? And so this is showing like histone with this tail sticking out, and all of these are available sites to allow for modification. That's got to change the conformation of those histones. Okay, and then that will then lead to a much more long-term change in, say, a gene being turned on or a gene being turned off. And it's this kind of process that occurs that allows for like cells to differentiate or for cells to change their gene expression based on, well, in the brain, it can lead to changes in like genes being turned on that allow for memory formation to have occurred, those kinds of things. So, okay, I don't think that we've discussed microRNA in this class yet. So we've talked about mRNA, right? mRNA is, what does that do? What do we use mRNA for? What does that M stand for? Messenger. Good. Okay. So what does that mean? Like where in the dogma, the central dogma for biology, where does mRNA fit in? What role does it play? Yeah. Yeah, essentially right. It's kind of the in-between stage from the DNA code to the protein, right? We transcribe mRNA, and then we use that mRNA as a way to translate the code into protein. Exactly. So that's the role of mRNA. And we normally think of, oh, well, that's, RNAs that get generated obviously must do that. Well, that's not the whole story, it turns out. So there are other categories of RNAs that are non-coding. So they don't actually code for proteins. They get made, but they do other stuff, it turns out. And we're learning more and more about this. There's still a lot of stuff that we don't know about this, but one category is known as miRNA or microRNA. These are very small segments of RNA that gets produced. So they get transcribed, but they don't translate into anything because they're non-coding. They don't actually make proteins. And they tend to be short, 20 nucleotides, 30 nucleotides, 40 nucleotides. So it's kind of too short to make much of a functional protein anyway. They don't have 5' caps. They don't have 3' poly A tails. They are missing a lot of those things. They do, though, form complexes with other proteins that are produced that are known as RISCs, so RNA-induced silencing complexes. So these are protein complexes. And the RNA that is made, this miRNA, may be complementary to another mRNA that is being produced. So the mRNA that is being produced, when it binds to this RISC complex, it will then bind to, in a complementary way, to that mRNA, Now that mRNA can't be translated because it's got this big honking thing attached to it, and this RISC complex targets it for degradation. So if you have, this is a way to shut down mRNA that the cell is wanting to shut down. So mRNA can be degraded slowly over time. It generally will take days to shut it down. But if you turn on a microRNA that is complementary to that, you can then shut it down very, very quickly. You can eliminate most of that mRNA in a very fast fashion in a matter of hours, basically. So this is a way of shutting off gene expression in a, sort of an epigenetic way. This is above the genome. So there are other kinds of non-coding RNAs referred to as long non-coding RNAs. So these are not micro. Instead, these are RNAs that look a lot like mRNAs in that they have 5' caps. They have 3' poly A tails. So these are the things that are going to allow them to persist for a long time. They even have introns. So they will have exons and introns. But unlike mRNA, long non-coding RNA lack a reading frame that allows for amino acids to be made. They are non-coding. They don't make a protein. So what the heck are these? And these tend to be hundreds to even thousands of base pairs long. So these are long, much bigger than the microRNAs. They appear to be involved in chromatin modification. And so we're going to talk a little bit about what long non-coding RNA does. Now, they fall into 4 categories. The textbook spends some time on this, and I don't understand quite why it's so important, but let's go through them anyway. But basically, long non-coding RNAs can be like on the complementary strand, and therefore it'll be antisense to a gene. So this would be a protein coding gene, and the long non-coding strand might be antisense to that. So it's related to the sequence, but it doesn't even necessarily mean that it actually regulates this, but potentially it could. There's also long non-coding RNAs that are intronic. So they'll be in the introns or between the exons of a gene that actually codes for a protein coding gene. They can be bidirectional in that, like, you may have within the genome a protein coding gene, and then next to it a gene that makes a long non-coding RNA that goes in the other direction, that is read in the other direction. And then they can be intergenic, of course. So therefore, sometimes you find them in between genes, which where else do they got to be other than between genes? I don't know why that's important. Whatever. All right. So the thing about long non-coding RNAs, like I said, they tend to be long, and they tend to make these complex structures where they can loop on themselves and have sort of almost a helical structure to them. Okay, but then they'll have these long tails that are coming off of them as well. Now, the role of these, we're still learning more about them, and there's evidence for each of these models, and they probably all do different long non-coding RNAs probably do contribute to each of these models in some circumstances. But let's just kind of walk through each model. So one model is the decoy model. So in this case, we have the long non-coding RNA that, let's back up here. So you may have a protein, say a transcription factor, that is binding to a promoter region within the DNA, okay? And then this is then going to drive gene expression downstream. If you are expressing a particular long non-coding RNA that is complementary to this protein sequence, so it'd be similar to this sort of nucleotide sequence here, this long non-coding RNA is kind of more freely available than this, and therefore that transcription factor may bind to the long non-coding RNA instead of the protein, instead of the DNA. And this therefore then serves as a decoy for those transcription factors. So this is a way of turning down gene expression by having a decoy that the transcription factor binds to. So this is one well-known process by which long non-coding RNA can affect the transcription of other genes. So it serves as a decoy. The other thing is that it has these other unique attributes to it where it can act as sort of an adapter for multiple proteins. So it can act as this platform to bring DNA binding protein complexes and sort of either be as a way to sync for them. So it'll act as a way of like, you don't need to be going into the nucleus and modifying the DNA, come bind to me. And so therefore it can act as a sync. It also can be a guide. So as I said, it has these sequences that may bind to certain proteins, say chromatin modifiers like methyltransferases or other things. And then it'll have these tails that may be complementary to a portion of the DNA. So then it'll bring these complexes to the DNA, where then the proteins now are targeting a certain region for, say, methylation or demethylation. So it's bringing complexes to these sites. So the long non-coding RNA can act as a way of mediating epigenetic change to the genome. Does that make sense? And then you have these sites that are then complementary to a portion, so that it gets targeted to a particular place within the genome, right? So you can see the structure here where it binds to a certain part of the genome, and then you have this other part here that is complementary to the proteins, so that it's brought into the genome and does stuff to it. The last thing is that it can act as an enhancer. So some long non-coding RNAs have these long, sort of very complex 3D structures, where they may bring in a multitude of proteins into the nucleus, where it may bind to one portion of the genome, bend it in a way where now the gene is now open and available, and then the other proteins that it's bringing in allows for greater transcription of that gene. So it can act as an enhancer, and this is known as an enhancer loop. And so it looks like long non-coding RNAs can do this kind of thing as well. Okay. So, yeah, questions on that before I go on? Yeah. So adding more complexity to the whole mRNA thing, in that now there are RNAs that don't actually code for protein, instead they do other stuff themselves. So great. All right. So now we're going to talk a little bit about how we can get monoallelic gene expression through epigenetics. Okay. So when we're talking about monoallelic gene expression, this is where we have only one allele that's transcribed, while the other allele is actually transcriptionally silenced. So it's not that like there's a difference in the kinds of proteins that are made, like one allele just is more dominant than the other, and therefore like it creates a thing that does more stuff, and that's typical Mendelian inheritance. Instead, we get monoallelic expression because one allele may be completely silent because of epigenetics. So there are three major classes of monoallelic expression, okay, because they're nine. So one is parent of origin monoallelic expression, or imprinting, which we're going to talk about in a moment. The next is random monoallelic expression, which is done through inactivation of the X-chromosome. So we've already talked about that, but I'm going to go into that in more detail. And then the last thing is random monoallelic expression of autosomal genes, which is similar to X chromosomes. So let's talk about imprinting. Imprinting is where you have genes that show expression from either the maternal allele or the paternal allele due to the fact of whether a site is methylated or not. So basically, this kind of parent-specific pattern forms during the formation of gametes. So as the gametes are being made, you'll get methylation that'll occur or not. And then if you have differential methylation of CPG islands, this will then produce an allele-specific pattern. So in this case, we're looking at two genes here. We've got IGF2 and an H19. And in this case, the paternal allele is methylated, and therefore we get reduced expression of H19. This allele from dad is not being expressed. But the allele, the strand from mom, that CPG island is unmethylated, and therefore we get high expression of her allele. So therefore you have monoallelic expression. And then there's a different mechanism by which we get like silencing of IGF2 on mom, but high expression from dad. Okay. So you can lead to this methylation and epigenetics leading to regulation of certain patterns that is imprinted from one parent, and therefore you get one allele expressed from one parent and not from the other. Okay. This also has effects on the X chromosome. There's interesting stuff about that, but I don't have time to get into. But I wanted to talk about a particular genetic disorder that is related to imprinting, and this is referred to as Beckwith-Wiedemann syndrome. This is one of the classic imprinting genetic disorders on chromosome 11, okay? And it involves these two genes we just talked about. So this is the normal arrangement that typically occurs. And in Beckwith imprinting disorder, what will happen is that instead of one allele being repressed through methylation of the H19 gene, both of them are repressed. So you have reduced expression of H19. So the H19 alleles are silent. And then this also means, and in this case, we get BWS if the IGF is also highly expressed across both alleles. So you have too much IGF being made and not enough H19. And this results in all kinds of abnormalities in the growing embryo. So it leads to like enlarged tongues, oral facial changes, stature changes, cognitive impairment as well, all because not a mutation in the genome itself, but only because there's been too much methylation of one gene. So it's not even that there's a mutation of the genome, it is that we have sites that have been highly methylated on both chromosomes. And if it results in high methylation of both chromosomes, well, then we get this abnormal pattern of IGF-2 and H19 expression, which results in malformations of the fetus, which is wild to think about. It's not even a mutation in the genome itself. It's just too much methylation. So, okay. And there are other forms of imprinting and epigenetic disorders discussed in the textbook. So one thing that also happens is that in mammals, there's reprogramming that occurs with every generation. So when the gametes begin to form, there tends to be a lot of a removal of methyl sites and other epigenetic signatures on the genome. And this is referred to as reprogramming. So it tends to get reset. Now, if it's reset completely, there's debate about that. It does appear that there can be transgenerational effects, which we'll talk about in a moment. And then it would have to pass through gametes if that's the case. There's a lot of debate about it. I tend to err more on the side that I'm not a big believer in transgenerational effects, but there are some evidence that indicates that it does seem to occur. So the reprogramming occurs in the parental germline as in the developing embryo just before implantation. So there tends to be a bunch of the removal of these sites. And then after implantation, you get differential genomic remethylation that recalibrates then the maternal and paternal alleles that'll be activated or inactivated. And so that one will be turned on, one will be turned off, not just on sex chromosomes, but also even within the autosomal chromosomes. Now, speaking of sex chromosomes, so we've talked about inactivation of the X chromosome, and a big part of what determines whether one X chromosome, either from mom or the X chromosome from dad, that gets turned off in genetic females, is that that's kind of assigned randomly, but a big part of it is that it's determined by methylation. So the X chromosome that is highly methylated, and that happens sort of randomly during the course of development, that'll be the one that gets turned off, okay? And then that ends up as a bar body. If you remember, we talked about bar bodies and inactivation in the X chromosome. This happens randomly over the course of development in mammals. So as an embryo is growing, it might be that there'll be a cell here where the X chromosome from mom gets turned off, and then a cell over here where the X chromosome from dad gets turned off. And then after that, the cells that emerge from those developing cells as they're dividing, they all stay the same. So all the daughter cells from the one where mom's X chromosome is turned off, all of them will be X chromosome, just dad being expressed. And then the one where dad is turned off, it'll be just mom that's expressed. So you get this mosaic pattern from that inactivation that occurs. And this is one of the reasons we see mosaicism in things like calico cats. So calico cats are only found in females for the most part, with rare exceptions, and you don't see this in male cats. And this pattern is because in the course of the development, we know that coat color in cats is found on the X chromosome. And so in females, if you have say an orange hair color that you get from dad, and then you have a black hair color that you get from mom, this inactivation pattern that's happening developmentally means that early on in development, you get the inactivation. So there was some cell here that allowed dad's orange hair to be expressed. And so then that ended up as all those cells that were born from that ends up as this chunk of orange hair at Gesundheit. And then from mom, there might be, so dad's orange hair is suppressed, but mom, say, has a black hair gene on her chromosome. And then all the cells that are born from that cell, that inactivation event, they end up just producing black hair. And that's why you get the mosaic pattern on calico cats in females. You don't get it in males because they only have one X chromosome. Does that make sense? Now, rarely it does occur that you do get a male that has a calico pattern. Does anyone know why? Why would that happen? Yeah. Yes, exactly. So there's cat version of Klinefelter syndrome. That's exactly what it is. Yes. And so they have two X's. And it's rare, but it does happen. Yeah. Exactly. So, all right. This kind of pattern of activation, inactivation, methylation, demethylation can result in mosaic patterns of expression in a developing fetus, in a juvenile, and in an adult. And so basically you could start with a cell where, say, both alleles are turned off for one particular gene because both sites are methylated. As the cell is dividing, maybe there's a daughter cell in which both sites become unmethylated. And now we have both alleles that get expressed. So you can have two alleles that can be expressed. And then maybe we have some other cell divisions from other daughter cells where either, say, the maternal site is unmethylated or the paternal site is unmethylated. And therefore, you only have one allele expressed. Or we might have a different daughter cell where both sites continue to be methylated and you get no expression whatsoever. So within each human being, you get this mosaic pattern of expression of alleles if we're talking about methylation during the course of development. And so we get a weird tapestry of expression even within an individual, not just with X chromosomes, but also across the autosomal chromosomes. And so you may have parts of you where you have a high expression of a gene that you got from dad, but another part where you have high expression from a gene that you got from mom. And it's because of this methylation randomness that occurs over the course of development that leads to this. Does that make sense? Okay. So methylation is also really important to cancer. And it turns out that hypomethylation is a major property that is observed in all cancers. So when we look at cancerous tissue, if we look to see, well, how much of the genome is methylated, we find overall that it is lower methylation than what we would expect. And what that means is that there tends to be a lot more genes being expressed than we would normally expect. And that aberrant expression of lots and lots of genes tends to do bad things to cells because lots of genes are involved in stuff that cells don't necessarily want to be expressed, such as like high proliferation or cell survival or other things that we're now having a difficult time regulating because they've been demethylated. So if they're demethylated, we get an expression of a lot of crap that we don't want in these cells. And so with cancer cells, they tend to have a lot of stuff that gets expressed. So overall, they tend to be hypomethylated. Does that make sense? Okay. But not all genes. In fact, there are certain categories of genes known as tumor suppressor genes that tend to be hypermethylated. So we find that these genes, they tend to be very important for cell cycle regulation, DNA repair. Many cancerous tumors, these genes will be overmethylated when you compare them to healthy tissues. So because they are hypermethylated, these important genes that keep cells in check, they are now, they have diminished expression because they can be hypermethylated. And we tend to find that in cancers as well. So let's talk about this a little bit of the important role of tumor suppressor genes. So we can get things like epimutations. So you can refer to the incorrect methylation of a gene as an epimutation, okay? That is one way to refer to it, versus just a genetic mutation where the genome has been changed. And it tends to happen in two stages. So as a cell lives for a long time, or as a person lives for a long time, and their cells, of course, are living with them, hopefully, you may get a mutation that'll occur in the genome of, say, a tumor suppressor gene. Well, the good thing is that they have two copies. So just because we have a broken version in one copy of the genome, we have the other allele that can serve as a way to continue to protect the cell. Or maybe they've had an epimutation that's occurred. So in this gene, perhaps they've had a methyl group that gets added that now suppresses the gene from being expressed on one copy of the allele, one allele copy, but we have the other allele and it's fine. So that's the first event. This is like not good, but everything is okay at this point. Now, if the cell experiences a second event, that's where things can go haywire. So if we look at this, it could be that this cell could then also have another mutation on the complementary allele, or that it's lost through some sort of translocation event, or that it gets methylated through an epimutation. Well, now we don't have any viable copies. So we're not getting any production of, say, the tumor suppressor gene. And therefore, now we can't suppress tumors at this point. If we start with an epimutation, well, you could still have these same things occur, right? You could get a genetic mutation, you can lose the gene on the complementary strand, or you might have another epimutation that gets added. So now you can even have just 2 epimutations leading to a tumor suppressor. So you don't even need to have a mutation in the genome itself. It's still leading to cancerous formation, which is not great. Now, this is a list of some of these tumor suppressor genes that are known to be sites of hypermethylation that lead to cancer. Some of these genes are some of the most famous genes in the genome, like BRCA1 is a breast cancer related gene. This gene makes a protein that's involved in DNA repair. So if people have a certain mutation in this gene, they have very high risk of breast cancer and ovarian cancer. And that's because they need that protein to make DNA repair so that the cell doesn't accumulate additional mutations where it's then going to have aberrant gene expression, including the expression of genes that promote cancer, which are known as oncogenes. So these hypermethylated genes within this list, they tend to be involved in DNA repair, differentiation, apoptosis, which is programmed cell death, related to drug resistance as well, like for anti-cancer drugs. So basically, cancer is now viewed as a disease that involves both epigenetic and genetic changes that lead to alterations in gene expression. Okay, so this is one way of thinking it. So we do know that in the promoter sequence that we have the CpG islands. If they are hypermethylated on tumor suppressor genes, well, then that's going to lead to these genes to be turned off. And then these tumor suppressor genes, they're heavily involved in also preventing genetic mutations. So if these tumor suppressor genes get turned off, well, now we're going to increase the risk of more genetic mutations, which is then going to maybe mess up some of the tumor suppressor genes, which is also going to lead to, say, oncogenes that get turned on, perhaps through changes that allow for hypomethylation to occur. So the hypomethylation leads to the oncogenes to being turned on, and hypermethylation leading to tumor suppressor genes being turned off. And then you throw in the addition of all these genetic mutations. Well, now you have the recipe for cancer to be formed. And it's thought that methylation plays a major role in this. So yeah, this is basically just a summary of everything I just talked about. That we have global hypomethylation that seems to underlie cancer, but we have epigenetic mechanisms that can replace mutations as a way of silencing genes. So you don't even need to have like a genetic mutation. You can just have an epimutation. And then epigenetic modifications can have an effect on normal cells, turning them into malignant cells through subsequent mutations because we've now modified DNA repair mechanisms. Does that make sense? Okay. All right. So I've got one last thing I'm going to talk about, which is how we can get environmental induction of epigenetic change. So it is also known that this changes to the genome, these epigenetic changes, can occur through environmental effects, such as effects on nutrition, chemicals, physical factors such as temperature, or other things that can affect the epigenetic state of the genome, the epigenome. Now, one line of evidence for this comes from studies shown that women who were pregnant during the 1944 to 1945 famine in the Netherlands during World War II, they gave birth to children that had a much greater risk of obesity, diabetes, and coronary heart disease if the woman was experiencing famine during that time, during pregnancy. And this was a lifetime effect, right? So they were monitoring these individuals born 40, 50 years later, and they had much greater risk of coronary heart disease. There's even some evidence that generational trauma can be passed through the epigenome, including even with follow-ups to these studies that you can have abnormal patterns of weight gain and growth, even in the F2 generation. So not just the F1 being affected. So we'll talk a little bit about how that can be passed on, but let's talk through a particular study. So I'm going to skip that one slide. So basically, so we're going to walk through one study that's very famous. This is a very famous study that showed that maternal care can have a huge effect on behavior in an adult animal. Okay. So when they get raised as a pup, it can have these lifetime long changes that can occur. And they describe the mechanism in the study. So we're going to go through this. So stress-induced epigenetic changes that occur prenatally or early in life can influence adult behavior, okay? Basically, there are some rat moms that show high levels of maternal nurturing. This comes in the forms of lots of licking and grooming. And so they refer to these pups that are born to these moms as high LG. And then there are other moms who are not very good at doing maternal care. So they have low licking and grooming rates. And this is referred to as low LG. Okay. So there's some moms that are just really good moms and some moms that are not so good amongst rats. Okay. The other thing that is the premise for this study is that they looked at the promoter region for the glucocorticoid receptor in rats. And in the promoter region, they found a CpG island. And within that island, there were 2 CpG dinucleotide sites that they were particularly interested in. And they refer to this as 5' and 3'. And they were really interested in the 5' region. So there are a number of other CG sites. As you can see, this is the CpG island. And I took this figure straight from this paper. So if you want to look at this paper, you can look at the link. All right. So what are some things that they found? So they had pups that were raised by either high LG moms or low LG moms. And then they sacrificed those pups and they killed them at either embryonic day 20, postnatal day 1, postnatal day 6, postnatal day 21, or postnatal day 90. Now, embryonic, obviously, well, they're not pups at that point. Postnatal day 1, 6, and 21, like this is sort of late stage juvenilehood. This is when they're basically ready to wean, or they probably are weaned at this point. And then P90, these are technically adults. So a P90 mouse or even a rat can reproduce at that point. So what they found is that if they looked at that 5' CpG dinucleotide site, that methylation in the hippocampus, so this is in the brain, was really high at postnatal day 1. But by postnatal day 6, high licking and grooming demethylated the site. You can see that the methylation is very low at the 5' site. But if there was low licking and grooming, so if there was with this kind of mom, these pups continue to have high methylation, nearly 100% methylation of all the sites, okay? Whereas high grooming leads to demethylation for this glucocorticoid receptor expression, okay? They found that this isn't a genetic thing. So they did cross-fostering. So obviously with low licking and grooming pups given to low licking and grooming mothers, they have high methylation. High licking and grooming pups given to high licking and grooming mothers, so like a pup that was born from a high licking and grooming mom, given to a high licking and grooming mother, low methylation. But if you take a high licking and grooming pup, so it was born to a high licking and grooming mother, and you give it to a low licking and grooming mother, so it's raised by one of these moms, you get high methylation. So we're able to, so it's not related to like the biological parent, it's related to how they were raised. And if you take a low licking and grooming pup, so you take one of these pups and you get it raised by this mother, well, they get low methylation, okay? So you can reverse the process by just doing this cross-fostering. So it's about how they're raised, not who they're born from. Does that make sense? Okay. All right. So, okay. What is then the consequence of this? So great, we got this methylation difference. It turns out it's pretty substantial. So they also looked at in adults the expression of glucocorticoid receptor in the hippocampus. And if you have pups that were raised in a high licking and grooming environment, you have higher expression of glucocorticoid receptor in the hippocampus relative to pups that were raised in a low licking and grooming environment. So pups that had the bad environment have half the expression of glucocorticoid receptor in the hippocampus relative to those that have high expression, okay? And glucocorticoid receptor expression in the brain is related to their ability to deal with stress, because this is related, it is the signal that corticosterone binds to in the brain. And so if you have high levels of expression, that tends to be associated with managing stress better. So we'll get to that. One thing that they also did to test this, to see if like they could reverse this, is that they infused a chemical called TSA. And if they infuse this into the brain in an adult, this reverses the 5' methylation, which they have other parts of the study that shows this. This leads to an increase in glucocorticoid receptor expression in adults that were raised by low LG moms. Okay, so in this case, they either infuse vehicle, which is here, or vehicle, which is here, whether you're LG or HG, okay? LG, low versus high. So licking and grooming the low, or high licking and grooming here. So the low licking and grooming, obviously they still have low levels of expression, whether you infuse vehicle or not, of glucocorticoid receptor. If you put in TSA into the brain to reverse the methylation, you can increase the expression of glucocorticoid receptor in the hippocampus in pups, or these are adults that were raised by a low licking and grooming mother. So you can reverse this methylation process, resulting in greater expression of glucocorticoid receptor. Okay, does that make sense? So we were able to take low licking and grooming pups and convert them into having high expression of glucocorticoid receptor in the hippocampus by infusing this chemical that reverses methylation. Okay, so it becomes demethylated. And of course, the high licking and grooming pups, whether you do TSA or not, it doesn't really matter because they already had high levels of expression of glucocorticoid receptor. So now we got 3 categories of animals that have high expression of glucocorticoid receptor and 1 category that has low. One of them was raised by a low licking and grooming mom, but 2 of them were raised by high licking and grooming moms. And then of course, 1 didn't get the reversal. It has low licking and grooming. So then the last thing that we're going to look at is of the data from this paper is their ability to handle a stress response. So if you take a rat and if you restrain it, it will not like that. It will have a stress response. And so there'll be a release of corticosterone into the bloodstream. The higher that level of release of corticosterone, the more prone that animal is to being very highly stressed and to also have other deficits. As I mentioned, there were 4 groups that we had, and we have 1 of which that had low levels of glucocorticoid receptor in the brain. And those are those individuals that were raised by a low licking and grooming mother. And you can see that they have a much more robust corticosterone response, that even before restraint, they already had elevated levels of corticosterone, right? So already, even before they did the restraining, their basal levels are already high. So they're already just kind of stressed. But 10 minutes later, 20 minutes later, 40 minutes later, 60 minutes, or 90 minutes later, after they do the restraint, you can see that they have a much more robust stress response compared to all the adults that were raised by high licking and grooming mothers, as well as the pups that were raised by a low licking and grooming mother, but had the methylation reversed in adulthood with the TSA infusion. Does that make sense? So we're able to reverse this process. But also, those that were raised by a low licking and grooming mother and had high methylation and low expression of glucocorticoid receptor, they have elevated stress response. Now, the idea behind this is that this can then lead to transgenerational effects in some ways. So if you have a low licking and grooming mom that raises pups that now have methylated sites in the brain, leading to lower expression of glucocorticoid receptor, as adults, they tend to be stressed out and not very good moms. And they also tend to not do very good licking and grooming of their pups. And therefore the cycle repeats. And so therefore those pups end up as low licking and grooming moms as well. So that's one way in which we can get transgenerational effects through an epigenetic mechanism. So it's an epigenetic mechanism that's allowing this to occur just because the cycle is repeating itself. Whereas with high licking and grooming moms, there's a lot of serotonergic drive, which is one part of the mechanism, which I didn't get into. It leads to these sites being unmethylated. So you get high expression of the glucocorticoid receptor. These are, as adults, they can handle stress. They tend to be kind and comfortable. And then they also handle the pups quite well. And they do lots of licking and grooming, leading to repeating of that cycle. And that is how you can get transgenerational effects. So, any questions on this? Obviously, I spent 10 minutes talking about this, went through it in quite a bit of detail. I would take note of that. Okay. Right. I didn't just tell you this for fun so that you can go home and ask your mom to lick and groom you when you go home for Thanksgiving. Yes.
So, not like you said, the plastic is
Yeah.
but what
So this mechanism, I don't doubt. I think that this is definitely the way it occurs. Now, there are some people that speculate that some of these transgenerational effects are actually happening through the germline. And that, and so there's more debate about that somehow, because I think the cross fostering also shows that it's not happening through the germline, obviously, right? Because you can take a pup that's raised by a low-looking and growing mom, or that was born to a low-looking and growing mom, have it raised by a high-looking and growing mom, and it has high methylation. So, but there are other studies that have shown like transgenerational effects that violate the whole fostering, and I don't fully understand how that can occur because of the resetting that occurs when an embryo is born, right? A lot of the sites get demethylated. So how do we have that effect that continues on? So that's the part that I don't get. So, but as far as this, I don't have any skepticism about it. Yeah. Which is why I wanted to spend 10 minutes talking about it. Yeah. Because it's a good study.
Have a good break.
Yeah, you too.